Dr. Vockley is probably the best metabolic physician in the world, and is very active and is based in Pittsburgh. And at the moment, today, there are no clinical trials in PKU taking place at any of the clinics across Canada, any of the 21 clinics. Dr. Vockley is going to talk to you about several active clinical trials, plus one or two others that are in the pipeline in the foreseeable future, as I understand things. And so it strikes me that Dr. Vockley's standing in the community of metabolic treatment and research is such that the developers of new therapeutic ideas want to make sure that Dr. Vockley is on the team taking things into the clinic for trial purposes. All right, so, and with that, and Dr. Vockley has a soft spot for Canada, Canadians, and CanPKU, and that's been proven many, many times. So, and Pittsburgh's not that far.
I'll certainly agree with the latter statement, John. Your earlier hyperbole was appreciated, but probably over the top. At any rate, let me get my slides up here.
Now, do I have to do something about share screen or anything like that?
No, I have that.
Okay.
And as I said, I'm going to have a phone call here. So if you'll give me a second, okay. I'm gonna put myself on mute and take.
Okay. So we'll all be patient. Maybe I'll stop the recording for a moment.
Disabled screen sharing. So you do need, John, to under share screen, the bottom of your screen, you need to enable multiple presenters.
Right. Okay. What did I?
It should be the green icon in the middle bottom of the window.
Yeah. Did I tell you I was colorblind? Okay.
Well, it's the only one that isn't white, so it'll look dark to you then.
Okay.
It also says mysteriously share screen underneath it. Gotta give him grief.
I've clicked on share multiples.
That should do it. It did. All right. And you should have my screen now. Okay, so, excuse me, I'm also dealing with allergies here, so if I cough a little bit, that's what's going on. We're going to be talking about clinical trials for PKU, and I thank John for the opportunity to present this. Coming south of the border for a clinical trial is a little complicated, but we think we're trying to make it, well, we know we're trying to make it easier. We think we are making it easier. So if anybody ends up being interested, we'll certainly be willing to talk to you about it. And we can figure out what that looks like. So just my disclosures.
One person who couldn't make it because of a shift change is actually participated for 4 years in a clinical trial in the United States for Palynziq, including the 3-dose regime, including 2 years of most of the 2 years of the pandemic. So he was crossing the border for clinical trial purposes every 60 days throughout the pandemic. And the only time things went wrong is when they tried to save him a trip and shipped it. And the Canada border guys kept the product, which needed to be refrigerated in an unrefrigerated situation, and the drug spoiled. Right. So that's, by the way, that's just, so it's doable. Is my point.
It is doable, and I'm not going to spend any time on introduction because you all know more about PKU than I do. But I do like to point out to the physicians that I talk to that this is not a disease that is cured. We're able to control the Phe under the best of circumstances, And under many, and I would have to say most circumstances, that's very, very difficult as folks get beyond their early childhood years. So you all know that currently in the treatment of PKU, we have two drugs. We have sapropterin and we have pegvaliase. Sapropterin is the pharmacologic form of biopterin, which was approved in the States in 2007 and later in Canada. And it is, well, we'll talk just a little bit more about that in a second. And then pegvaliase, which is not yet approved in Canada, as I understand. Is that correct, John? So we have approval here in the States for 2018, but there are some trials going on for it, so I can talk to you about that. The role of sapropterin is relevant because one of the drugs that we're going to be talking about that's in development may be a competitor, if not a replacement for it. And about somewhere between 25 and 50% of the patients with pH deficiency or PKU have some response to sapropterin, depending on the study that you look at. If you have a mild or moderate disease, you're more likely to respond. And notably, some stable protein is required for sapropterin to function. So if you have one of those mutations that completely eliminates the protein, you won't respond to this. Excuse me. In the U.S., the current treatment guidelines recommend that everybody should be given a trial of sapropterin first, unless you have two mutations that completely knock out protein, and then that if you don't respond, then you go on to the other modalities. Now, pegvaliase is a very interesting drug because unlike sapropterin, which works a little bit for some people, but only normalizes a very small fraction of the patients who use it. Pegvaliase has the potential to normalize. Oops, somebody else giving me a message here. Has the ability to potentially normalize Phe in everybody, no matter your mutations or your starting Phe level. And this is just a long-term phenylalanine level that in the first, I don't know, this was about 80 patients on the drug, and you can see that the average starting Phe was 1,400. And the average Phe at the end of the study was about 420. And then individuals have had as low as 0, which we don't recommend, but you can get into a normal range with this drug. Now, the only clinical trial that's currently going on in the States, since it's already approved, is looking is looking at lowering the age range. So right now, it's approved to 18. Most insurers in the U.S. will cover it down to 16 because there have been some trials with that, even though it's not FDA approved for those ages. So the current trials are looking at will ultimately be looking at younger patients, starting in the late childhood and early teens and working on down. I do not think we have the option of bringing adults into any of the trials that we have for pegvaliase. So keep that in mind as you're looking at it. One of the reasons that pegvaliase isn't the only drug we need is that it's kind of hard to use. It's a daily injection or 3, depending on your response. And there's a fair amount of hypersensitivity reactions. And these reactions, as you can see here, go out over time and can show up anytime in the first couple of years using it, though it is more frequently seen in the first 6 months. And those can be severe enough to be called anaphylaxis, requiring an emergency room visit. So it works, but it's not the easiest drug in the world to use. And as a result, there's still a lot of development in progress, working for new therapies, and we break them up into several categories. There are the nucleic acid therapies. This is gene therapy and mRNA therapy. There are cellular therapies. Currently, the only one of those that's active is ours for hepatocyte transplant or liver transplant. And then there are small molecule therapies that are mostly designed to help mutant PAH enzymes fold properly and restore enzyme activity, whereas otherwise those proteins just break down and are recycled. And then in the long saga of the development of a home feed eater, there is yet a new development, and we'll talk about that at the end. So there are two companies doing gene therapy. And if you look at the way this is done, I'm showing here some data and the vector that's being used for the homology program. This is what you do is you insert the PAH gene in the middle of a virus. And then package that into the virus. You put it in the middle of the virus genome, package it into the virus, and then you intentionally infect an individual with that virus. And if you can see here that in mouse studies, you inject the virus, and the phenylalanine level, which starts off at around 1400 or 1300 in these animals, goes down to normal. Their phenylalanine levels drop also down into the normal range, and the mice who have a light-colored coat because of the melanin defect in PKU develop dark coats. So this has worked really well in the mice. This just shows another image, a little bit bigger, of treating these animals and seeing that their levels going down for a long period of time. A year is middle age in mice. They'll sometimes live out to 2 years. And the first vector they used wasn't terribly effective, so this is the one that moved into clinical trials. So the mouse models look good, and the clinical trials currently in progress. There are, I think it's now 8 patients who have been treated in a dose escalation study. And there are some early signs of efficacy that the companies release, but they have not released a full report on those first that first cohort yet, or the first 2 cohorts. But this is active, and we have this trial in Pittsburgh. The one thing that you should know is that it does require some immune suppression in the early part of the treatment, and that's not very severe. It's prednisone. It's a steroid that keeps your body from rejecting or from reacting to the virus, because it sees this just as a regular virus, and letting it get into the liver, infect the liver cells, and now the liver cells are ready to go. BioMarin has got a vector that looks very much like this. Their trial is active, but not currently enrolling, and so there will be 2 of these. And we also have the BioMarin trial here in Pittsburgh. There's been a major concern related to AAV vectors, which is what this is, that has recently led to the FDA to call for an evaluation of the technique. There is a condition called thromboembolic microangiopathic vessel disease or hemolytic uremic syndrome. And what this is it's an immune response where the body starts reacting against blood cells and kidney cells. And so it can be very serious. You can end up on dialysis. You can end up needing blood transfusions due to bone marrow failure. And the thing is that we just don't know what causes it. In the homology study, no one has developed it. However, across the board, probably about a quarter to a third of patients who get an AAV vector develop it. And it's not clear whether it's related to the vector, whether homology's just been lucky, or we have to worry about this going forward. So it would be something we would talk about in detail if you were to be interested in this. The other issue that we have to worry about is that the liver is constantly replacing itself. So in kids, gene therapy with an AAV vector, which does not replicate by design, the liver eventually outgrows it. In an adult, that is a much more slow process, and we don't know whether this will last 5 years, 10 years, 20 years, or even a lifetime. But chances are pretty good that eventually the efficacy will decrease and you'll require another dose of something else. You can't get the same vector twice because you become immune to it. But for example, if you got the homology, you could eventually get the BioMarin, or there are 3 or 4 other companies that are developing PKU gene therapies. Now, mRNA therapy is being developed by a company called Moderna. You might have heard of them. They make mRNA treatments, and long before COVID was a thing, we'd been working with them on rare diseases. I work with another group of disorders called fatty acid oxidation disorders, and we've been building an mRNA for one of them, VLCAD deficiency. And this is just to show you that we can infect cells or a mouse, and we can get a huge burst of activity. And this is much less, it doesn't cause the reaction that the AAV viruses do, but it's also transient. So this does not get in there and stay in there. Most of the current therapies with mRNA treatment require an every 3-week infusion. This is the one trial that's active. So VLCAD's not active, PKU is not yet active, but this is one that's active, methylmalonic acidemia. And you can see that if you treat a mouse, you get something that looks pretty much like what we showed in the gene therapy. But then if you let this go for too long, it goes right back up to where it was before. This is it is sustainable long term. We treat patients with enzyme replacement therapy with a similar kind of dosing schedule every 3 weeks. So this is not out of the realm of possibility or even reality. But it is not. It is an ongoing therapy, and so there'll be some issues that are certainly related to that. Now, gene therapy is getting a new gene into the liver cells. We're taking actually 2 different approaches here. One is we've actually done a liver transplant on a patient with PKU who had been unable to control her PKU and wanted to have children and knew the risks of maternal PKU syndrome and decided to have a liver transplant because she did not respond to Kuvan and she got terrible reactions to Pegvaliase. So she had nothing left. She just couldn't keep up with the dietary therapy. So since PAH is a liver-based enzyme, not surprisingly, she's now cured. Her Phe's been normal for a year. But any procedure that starts off with step 1, first remove liver, is a little scary. And so we're taking a different approach, and that is instead of having to replace the whole liver, what if we could just give you liver cells with a normal PAH gene back? So this is called a hepatocyte transplant, and we currently have an NIH protocol to a grant to do this. This was the first patient that we've treated, and she was done at a lower dose. But you can see that her level, and now this is in milligrams per deciliter, so she started off at about 24 22, and she dropped to about 14. And we're further up in the dosing schedule than that now. And we're pretty sure we can get the Phe levels down to close to 600 or maybe even lower with this technique. It is a one-time therapy, but it requires two things that are a little bit scary to people. One is we do a little bit of X-ray treatment to one lobe of the liver to cause it to stop growing and let the new liver cells go in there. It's a low enough dose that it has absolutely no long-term effect on the body, and there's plenty of evidence of that, but some people worry about that. But the other side of it is it does require the same immunosuppression as a liver transplant. Now, that's a lot milder than the steroids that we're using for the gene therapy, but it's ongoing. The gene therapy, eventually you wean off of the steroids, and this is a drug called tacrolimus, and we've had patients on it for 40 years with liver transplants. You heard mention of Synlogic, and this is a company that is building a probiotic approach to treating PKU. Here, instead of giving you a phenylalanine ammonia lyase, the enzyme that's in pegvaliase to you via an injection, they build it into a bug along with a couple of other genes that help metabolize phenylalanine. This is an E. coli nissl, which is the same strain of probiotic that you would get if you went to the drugstore and said, give me a probiotic. I have GI symptoms and I feel like I need something. This is what they would give you. But the companies manufactured or built the PAL, another enzyme as well called LAAD, and then the ability of a transporter to make the phenylalanine levels inside the bug higher. And what they've done is they've built in a gene that keeps the organism from replicating. So there's no concern about this somehow or other getting out of control and taking over. So if you stop taking the drug, it's just gone in a day or two. And so the goal here is to take this in some fashion. It might be with meals. It might actually be in between meals. We don't exactly know yet. And you can see what happens. So the population that's currently being treated are those individuals with classical PKU and phenylalanine levels above 600 micromolar. I think the new protocol allows you to be on sapropterin, but you can't be on pegvaliase. And then they want a stable diet history, so you're not moving your diet along. And the endpoints of the study are to measure your Phe level and see if it comes down. But they also do what's called a tracer study. And I'll show you what that looks like later. And that actually allows to, using a whole body technique, to see whether or not your body is metabolizing Phe now once you've got that in place. So the first trial in PKU patients was pretty involved. It was a 30-day study where you took the dosing and then had some testing done at 2 different periods, plus at the end of the protocol, and then did this tracer study that I mentioned near the end. And what the tracer study is it's a phenylalanine molecule that's labeled with a natural isotope that's not radioactive. So an isotope is just a variant of an atom. So instead of a carbon atom that has a molecular mass of 12, this is a molecular mass of 13. It's just naturally occurring. The one you might have heard of that's radioactive is carbon 14, and this is not carbon 14. So this is completely harmless. You could replace every phenylalanine molecule in your blood with this, and it wouldn't make any difference. So you drink this stuff, and it's metabolized, and we can tell the difference between the phenylalanine or its metabolites that have the carbon 13 versus carbon 12, the normal carbon 12. So it allows us to measure this in your blood. Oops, this didn't work. I must have forgotten to change this one. The first phase is done, and in fact, this organism did demonstrate the ability to digest the Phe in the GI tract. There was a 40% in reduction of the heavy Phe after a meal challenge with this medication. There was a 20% reduction in fasting Phe across the whole population, but there were some patients that seemed to respond and some didn't. And we don't quite understand that, but if you look just at the individuals who responded, there was a greater than 20% reduction in their Phe. We might get a little argument from one of our attendees. The company concluded that it was generally well tolerated, and I will say that we had several patients who complained about it, but we're able to tolerate it. So whether that's generally well tolerated or just put up with it, they have not volunteered to go to the next level. So I think that probably speaks to that they didn't particularly like it. However, an optimized version of this drug has been developed that can be encapsulated so that it may be a little bit more, it might be easier on the GI tract as it gets to where it's going to go in a more gradual fashion. And it's got about somewhere between 2 and 3 times the capacity of the original organism. So we think it's going to be a better drug. And in the non-PKU studies, it does look like it's metabolizing Phe at a higher rate. Now, I mentioned the home Phe monitor. We've been working with now 4 different companies to develop a Phe monitor. The first 3 of them have dropped out because they couldn't deliver what they said they had already done when we first started working with them. And so now we're working with a 4th company, and they also claim they have one that works. And we're going to work to do a clinical trial using that Phe monitor to show that using the Phe monitor, you can better manage your Phe at home, that you're able to keep it lower by looking at by knowing your Phe level in real time. So hopefully that happens, but it's likely not to happen for another year, I'm told. So unfortunately, still waiting for that to come down the road. Slight detour, just because we're now working with a model of PKU that we've developed in a pig. There's that mouse model that I mentioned to you before, but it's not a very good model of PKU because it doesn't reproduce many of the symptoms of PKU, and the mouse metabolism is different from humans, and so it's hard to know how testing in a mouse is going to translate over into people. So we use CRISPR-Cas9 technology to knock the PAH gene out of a pig. And then you can see that these pigs have a very high level of phenylalanine, 2,000, compared to 120 for a normal. And if we put it on a diet, we can get it down to normal. And if we take it back off diet, it goes back up again. And so finally, we can titrate these guys down and keep them in a normal range. This is our affected animal, and this is our normal or heterozygote control. And you can see this is a smaller animal. This is the weight gain curve. It's smaller than the unaffected SIB through most of the first year. It catches up by the end of the first year. And the reason that I show you this is we're now working with Nestle to test yet a different version of PAL for treatment. This is an oral version. It is a an acid stabilized version of the drug, and it will be encapsulated. So it'll be taking a capsule, and much like that probiotic that I described to you. This drug should be able to digest the Phe in the intestine. They've shown that happens in normal controls, but in the controls, they were using an unencapsulated version of it, and it was digested rapidly, too rapidly in the gut to be useful. So now they've gone to this encapsulated version. So we're going to be testing this in our pigs. We think that'll take another year, and then this will be in clinical trials as well. The final trial that I'll tell you about is by PTC, I'm sorry, Therapeutics, and this is a drug called sepiapterin. And sepiapterin is a molecule that's in the biopterin synthesis pathway. So you start with the starting compound, and it goes through sepiapterin, and then it becomes biopterin. And sapropterin is a form of a final biopterin. And so the preclinical studies with sepiapterin have shown that it's more bioavailable than sapropterin is. It gets to higher plasma levels, not doses, than sapropterin, and as a result, there's a much higher percentage of patients who respond, and many patients respond with getting normal Phe levels in the phase 1 and the phase 2 trials. So we're starting a phase 3 trial, and here anybody, this is the only one that I can tell you that we have that is going to be active for children. Unless the pegvaliase trial ends up being a possibility for you. This will be for anyone over 12 months of age and with a Phe level of 360. You can't be on sapropterin, of course, and pegvaliase isn't an issue for you. And this trial is just getting underway right now, so it's active. I don't think anybody's been dosed yet, but most of the sites are getting up and running right now. So it's a dose escalation study, starts on low, you'll move up to a higher level, and the highest level, which we think will be the right level, will be the one that you'll stay on long term. Let me just take a couple minutes to remind you of what we're talking about when we're talking about a clinical trial. So clinical trials are broken down into phases. Phase 1 is really a safety study where we look at the drug metabolism and do some dose ranging, usually in healthy volunteers. In phase 2, you move to the target population. Still safety is an issue, but now you start getting the first look at efficacy. There may be some dose escalation here as well, and it may be blinded or it may be open label, and that just depends on the preparation. In phase 3, that's the big study that the FDA requires for approval. It's typically a randomized, double-blind, placebo-controlled trial, meaning somebody gets a placebo, a fake drug, and somebody gets the test drug. But in this case, eventually, once the main part of the study is open, everybody gets to go on the study drug. So the placebo portion is just a small part of it. And then once I forget how many we're looking to enroll in here, I think it's about 160 people. So once you get to that, they get the data and present it to the FDA for approval. In most rare diseases, there's what's called a phase 4 or a post-marketing study, and that is simply because you do so few patients in a rare disease clinical trial compared to a clinical trial for a more common disease, that the FDA requires companies to continue to collect safety and efficacy data after it's been approved for use. Some hints for participating in a clinical trial. You really want to be very, very sure you understand the purpose, both generally and specifically for your use. understand the logistics. Who's paying for the travel? Do you need to be in the hospital or is it outpatient? And then are there any procedures that need to be done? You hopefully know the investigators. Now, since your home teams aren't doing this, then you just have to find someone who can accommodate you in travel, and take the word of folks like John, who knows most of the investigators in the States and can tell you, yeah, they're a good egg, or nope, don't know them, stay away from them. And then always very carefully review the consent form and understand it, because it will call out all of the procedures, the potential risks, the potential benefits. It's important to know, and this will be in the consent, that you always have the right to refuse or withdraw without any consequences from a clinical trial. So, your home team will continue to treat you regardless. If the drug is approved and you want to use it, you can use it, no problem. And the most important thing that I like to remind people is that participation in a clinical trial is really a partnership between you and the investigative team that involves mutual trust. And obviously, you will want to ask your local team about it as well. Why do we need clinical trials? Well, it's the only mechanism for obtaining FDA approval for a new treatment, or treatment in Canada for that matter. There aren't any treatments for most rare genetic diseases. PKU is lucky, we've got 2 already. But if we don't get participants in clinical trials, we don't have any clinical trials, and we get no more additional trials. So the good news in PKU is that the number of drugs that are coming down the pipeline is really quite large right now. There's a lot of interest in it. There's some things that I didn't even talk about because they're just in the laboratory right now. But over the next 3 to 5 years, you can look to see probably a half a dozen different things going into clinical trials, and I think that it really. If you're not in a position to do these at home, most of these trials will be open to international travel, and so just contact me or somebody at whatever site is closest to you, and you'll be able to hear about it. So unfortunately, with all of the I'm going to stop my share with all of the interruptions. We only have about 15 minutes left, so I'm going to close up here and take the next 15 minutes to answer questions, but also let you know that you or anybody you know, anybody in CanPKU is welcome to contact me if you have questions about a clinical trial. John's got all my contact information. He could find me if I was on the moon. Just let him know and he'll get you in contact with me. I can also put it in the chat, but John's got it, so he'll just pass it on. So let's see. Is there any indication that the liver may regenerate without the virus? And if this is the case, can they be reinfected? Tanya, can I ask you to clarify that? Because I'm not sure I understand the question.
That's okay. I think you actually answered it in your presentation.
Oh, okay.
I typed it in. It was basically just the liver regenerating itself, but you mentioned children may be a problem, adults can
yes.
Reinfected.
I understand the question. Yes. So the liver is constantly regrowing itself. It's one of the only organs in the body that seems to have almost endless regenerative potential. And while in adults, you probably only turn over about 10% of your liver every year. What that means is likely in 5 years, you've cut the activity of the liver of the enzyme, the transfected cells, in half, and that's where the guesstimate of sort of 5 to 10 years is there. They're using CRISPR-Cas9 and gene editing therapy. You could actually get those liver cells to, when they divide, pass on the new gene, but we're not there yet. There are companies that are working on that, but it's a higher bar from an experimental standpoint. Let's see. Brian, you really did make a sour face there when we were talking about Synlogic. I don't know if you want to say anything else. Most people had GI symptoms. Uh,
I don't know what it was for me. I've gotten the okay to actually talk about this in a presentation that I did. I think that was in BC at the one of the Synlogic representatives was there. Who was it again, John? I can't remember his name.
That was Larry. All right. Yeah.
Larry,
Yep.
right. Yeah. Anyway, it was just, I don't know, things didn't go well. And number one, I if I taste that mint, that kind of mint now, I'm done. Like I'll be thrown up. And I ended up like I actually ended up throwing up and like, ended up having to, go through a little more than I think everybody else did.
So the hope is to get this into capsules, as I said. This next generation will hopefully be in capsules. Our other participants said the same thing, that it wasn't a pleasant experience to drink this stuff. So
it was a weird way to flavor it. I gotta say, like I did not understand why it went mint, but maybe it was masking something or something
I
like
think
that.
that's what it was. They got some taste testers around and they said, tell us what covers the taste of the medication the best, and they all said mint. But yeah, it maybe a little bit of a weird choice.
You don't often drink mint.
Minted ginger ale, come on. I grew up on minted ginger ale, but I think maybe this might be just a little bit stronger. Charles, issues with comorbidities for consideration. Anything in particular? The answer is nothing specific. Usually what it comes down to is if the investigator says whatever's going on with that individual, if you think it's safe for them to be in a clinical trial, it should be okay.
Okay.
Other questions about any of these trials or trials in general?
I'm getting a little older, and so is there an age limit where you would stop a clinical trial for consideration? Or
some of them do stop sometimes at 50. I don't think any. I think all of ours go up to at least 60. But I can look for you, and if there's any of these modalities in particular that you're interested in, then I can let you know.
Okay.
Or you can just Google it. Not Google it. Go to clinicaltrials.gov. I forgot to mention that. If you go to clinicaltrials.gov. That'll take you to the NIH site that has all of the clinical trials that are registered with the FDA in the United States, and it'll have the entrance criteria, the age ranges, all of that stuff will be there.
And for what it's worth, it also has a list of the all the trial sites.
Yep.
All right. So
yeah, you can find out who's doing it as well.
Geography matters to you. Is that what about? Yeah.
Dr. Vockley, I got to ask, is there certain trials that kind of like, if you do this certain one, it's going to exclude you from others, or?
So most of them, the general rule is that you can't be taking more than one experimental medication at a time. So if you're taking something and you continue to take it, you're done for the time being.
Right. If
you stop it, most of something like that's reversible. So the mRNA, the small molecule therapies, those kinds of things. There's a washout period, and then when the washout period is done, and usually it's 1 to 3 months, you can enroll in another trial. The two that'll be more problematic in that result, or in that respect rather, are the hepatocyte transplant and the gene therapy, because once you've injected those, they're kind of there, and so while it won't preclude you from taking the drug if it's approved, you can't just say, okay, all of a sudden I want to get rid of my gene therapy vector. You actually can say, I want to get rid of the liver cells. If you stop taking the immunosuppressant, the liver cells just go away. The body clears them off the way it does any foreign cell infection or otherwise. And you might have a little bit of a febrile reaction for a day or two, but then they're gone. And so I don't know of anyone who has come up with a timeline for use after hepatocyte transplant, simply because no one's doing hepatocyte transplants anymore besides us, so no one's paying it any attention.
Cool. Thanks.
I should have mentioned, I don't think I did, the hepatocyte transplant, we give you those cells the way you get a blood transfusion, so it's just an IV. So no surgery, no incisions, anything like that.
In summary, which are the trials that you are actively enrolling for now, and then which are the ones that, say, a year from now are most likely to be available?
PTC, the sepiapterin is enrolling. We are soon to be enrolling in the younger pegvaliase trial. We're doing I don't know why they continue to ask for the natural history studies, but they do. And then the 2 gene therapy trials are the ones. And let me just look at my list here and make sure that I got everybody else that I can't remember what all I mentioned. Oh, the Synlogic. We're enrolling in Synlogic. Don't want to forget that one. So that's Synlogic, sepiapterin, the PTC drug, the 2 gene therapies, and pegvaliase is soon but not immediate.
So just on practical logistics, if somebody enrolled tomorrow in one of those active ones, when would you be interested in starting therapy?
The PTC would be almost immediately, and any time thereafter. We have to wait for a place in the queue for the gene therapy. So it just depends on how many other patients are waiting in the queue ahead of you. So that could be anywhere from a few weeks to more likely a few months. The hepatocyte transplant could be almost immediately because we have no queue for that. The Synlogic, we're actually just in between trials right now. We finished the one that Brian participated in, and we're getting ready to start the other one, but that's going to be, in the next month or two. So sometime in the next few months for sure for that one as well.
Fairness to the audience, I should mention both. I'm aware that both Synlogic and PTC Therapeutics are in the process of finalizing 2 or maybe 3 trial sites at different centers in Canada.
Excellent.
That's not public yet, but it's also not something that's going to start imminently. So if anyone's interested in speed, then there is this option in the United States.
And keep in mind that if you start in one site, you can always transfer to another. So if you were anxious to try something and you wanted to start at a US site and one opens up closer to you in Canada, you could just transfer to that site.
Salt Lake City is lovely. Just saying.
It was gorgeous.
Hey, Pittsburgh's lovely.
Yeah, well, I'm hoping that there's a Pens game.
It's not the Rockies, but we got the Appalachians, and we got rivers.
And you got the Pens.
And we got the Pens. That's right, Sydney.
There we go.
I mean, come on, worth the price of admission. He's finally getting up to speed this year. He had a little slow start because of his injuries, but he's looking pretty good now again for an old guy.
For an old guy. Jeez, he's my age. Thanks for that.
In hockey, that's an old guy.
That must make the rest of you feel,
Okay.
Anytime I visit Halifax and I say I'm from Pittsburgh, I inevitably get into a conversation about Sydney. It's just,
Yeah. Guaranteed.
Right.
Yeah.
Okay. Unless there are any last minute questions, I'm going to say Thank you very, very much, Dr. Vockley, and allow you to get back to the campfire or however else.
Oh,
you're
got
trying to
another
keep
meeting
warm.
at 3 o'clock here. So yeah, it just keeps going.
All right, that's good. And we will keep in touch. And if there are, 5 minutes from now after the call, if the burning question enters your mind, let me know, and we can share it with Dr. Vockley and get back to you.
Happy to stay in touch. Happy to hear about questions on any of the trials. So thanks for being online, and for those of you who are going to listen after the fact, if you do have questions, just funnel them in through John, and he'll get them to me.
Okay. Thank you,
Thanks
Jerry.
all. Bye bye.
Thanks, Dr. Vockley.
Safe
Thank
flight
you.
there, John.
Take care. Bye bye.